MCQs, Recombinant DNA Technology. One who is considered the father of recombinant DNA technology. A. Kary Mullis. B. Paul Berg. C. Herbert Boyer. D. Stanley Cohen. Answer: B. Paul Berg. Paul Berg was the first to create a recombinant DNA molecule in 1972, combining DNA from two different sources. 2. Which enzyme acts as molecular scissors in recombinant DNA technology? A. DNA ligase. B. Restriction endonuclease. C. RNA polymerase. D. DNA polymerase. Answer: B. Restriction endonuclease. Restriction endonucleases cut DNA at specific recognition sequences, enabling gene splicing. 3. The discovery of restriction enzymes was made by A. Paul Berg and Stanley Cohen. B. Hamilton Smith, Werner Arber, and Daniel Nathans. C. Kary Mullis and Hargobind Khorana. D. Watson and Crick. Answer: B. Hamilton Smith, Werner Arber, and Daniel. These scientists discovered restriction enzymes and received the Nobel Prize in 1978. 4. The first recombinant DNA molecule was made using DNA from A. SV40 virus and lambda-phage. B. Adenovirus and E. coli. C. Human and yeast. D. Tobacco mosaic virus and E. coli. Answer: A. SV40 virus and lambda. Paul Berg used SV40 and lambda-phage DNA to make the first recombinant DNA. 5. Which of the following is the first recombinant DNA product approved for human use? A. Human growth hormone. B. Recombinant insulin. C. Interferon. D. Hepatitis B vaccine. Answer: B. Recombinant. In 1982, recombinant human insulin, Humulin, was approved by FDA, produced by E. coli. 6. The term restriction in restriction enzymes refers to A. Limited gene expression. B. Prevention of viral replication in bacteria. C. Control of DNA replication. D. Blocking transcription. Answer: B. Prevention of viral replication in bacteria. Restriction enzymes were discovered as bacterial defense mechanisms against bacteriophages. 7. Which of the following sequences is palindromic? A. ICT. B. ECTA. C. KAC. D. TACA. Answer: A. ICT. ICT. ICAR recognition site reads the same 5 rightward arrow 3 on both strands. 8. Which enzyme joins the foreign DNA fragment with plasmid vector? A. DNA polymerase. B. RNA ligase. C. DNA ligase. D. Restriction endonuclease. Answer: C. DNA ligase. DNA ligase seals nicks in the sugar phosphate backbone, forming recombinant DNA. 9. Which plasmid is widely used as a cloning vector in E. coli? A. PBR322. B. PUC18. C. TI plasmid. D. PSC101. Answer: A. PBR322. PBR322 is a classical cloning vector containing antibiotic resistance genes and origin of replication. 10. The enzyme reverse transcriptase synthesizes A. DNA from RNA template. B. RNA from DNA template. C. Protein from mRNA. D. rRNA from DNA. Answer: A. DNA from RNA template. Reverse transcriptase synthesizes complementary DNA, cDNA, from mRNA. 11. The first step in RDT is A. Ligation. B. Isolation of DNA. C. Transformation. D. Screening. Answer: B. Isolation of DNA. The process starts with isolation of donor DNA and vector DNA. 12. Enzyme used to remove RNA during DNA isolation. A. DNase. B. RNase. C. Lysozyme. D. Protease. Answer: B. RNase. RNase digests RNA, leaving pure DNA. 13. What is the function of lysozyme during DNA extraction? A. Degrades RNA. B. Breaks cell wall. C. Cuts DNA. D. Polymerizes DNA. Answer: B. Breaks cell wall. Lysozyme digests peptidoglycan layer in bacterial cell wall. 14. In recombinant DNA experiments, plasmid acts as A. Restriction site. B. Vector. C. Enzyme. D. Substrate. Answer: B. Vector. Plasmids carry foreign DNA into host cells. 15. Which of the following is not a cloning vector? A. Cosmid. B. Plasmid. C. Transposon. D. Bacteriophage. Answer: C. Transposon. Transposons are jumping genes, not used as vectors. 16. The enzyme used to amplify DNA fragments in vitro is A. DNA polymerase. B. TAC polymerase. C. Ligase. D. Helicase. Answer, B. Took polymerase from Thermus aquaticus, is heat stable and used in PCR. 17. The selectable marker in PBr322 plasmid is A. Orisite. B. Ampicillin and tetracycline resistance genes. C. Multiple cloning site. D. Promoter region. Answer, B. Ampicillin and tetracycline resistance genes. These antibiotic resistance genes help in selection of transformed cells. 18. The process of introducing recombinant DNA into bacterial cells is called A. Hybridization. B. Transformation. C. Translation. D. Transcription. Answer, B. Transformation. Transformation is the uptake of foreign DNA by bacteria. 19. The enzyme DNA ligase forms which type of bond? A. Hydrogen. B. Ionic. C. Phosphodiester. D. Peptide. Answer, C. Phosphodiester. DNA ligase forms phosphodiester bonds between DNA fragments. 20. Which method uses electric current to introduce DNA into cells? A. Microinjection. B. Electroporation. C. Gene gun. D. Transformation. Answer, B. Electroporation. Electric pulses create transient pores for DNA uptake. 21. Which of these is used for gene transfer in plants? A. Ti plasmid of Agrobacterium tumefaciens. B. Lambdaphage. C. Retrovirus. D. Yeast plasmid. Answer, A. Ti plasmid of Agrobacterium tumefaciens. Ti plasmid naturally transfers DNA to plant genomes. 22. In blue-white screening, recombinant colonies appear. A. Red. B. White. C. Blue. D. Green. Answer, B. White. Insertion disrupts LAC-Z gene, preventing beta-galactosidase formation rightward arrow white colonies. 23. Which of the following is not used in recombinant DNA experiments? A. Restriction enzyme. B. DNA ligase. C. DNA polymerase. D. ATP synthase. Answer, D. ATP synthase. ATP synthase is unrelated to DNA recombination. 24. Polymerase chain reaction, PCR, was invented by A. Paul Berg. B. Kary Mullis. C. James Watson. D. Stanley Cohen. Answer, B. Kary Mullis. Kary Mullis invented PCR in 1983 to amplify DNA. 25. The origin of replication ensures A. Protein synthesis. B. Replication of plasmid inside host. C. DNA ligation. D. Restriction digestion. Answer, B. Replication of plasmid inside host. Orisite allows vector to replicate independently within the host cell. 26. What is the purpose of a multiple cloning site, MCS, in a vector? A. Acts as selectable marker. B. Region containing many unique restriction sites. C. Provides antibiotic resistance. D. Helps in replication initiation. Answer, B. Region containing many unique restriction sites. MCS, polylinker, allows insertion of foreign DNA at specific sites using different restriction enzymes. 27. Which enzyme can cut both strands of DNA at specific recognition sequences? A. Exonuclease. B. Restriction endonuclease. C. Endonuclease. D. Ligase. Answer, B. Restriction endonuclease. Restriction endonucleases recognize specific sequences and cleave both strands. 28. Recombinant DNA molecules can be introduced into animal cells by A. Electroporation. B. Microinjection. C. Liposome-mediated transfer. D. All of the above. Answer, D. All of the above. Several physical and chemical methods can be used to transfer DNA into animal cells. 29. Which of the following describes sticky ends? A. Single-stranded overhangs created by restriction enzymes. B. Double-stranded blunt cuts. C. Unmodified DNA ends. D. Methylated DNA regions. Answer, A. Single-stranded overhangs created by restriction enzymes. Sticky ends help DNA fragments anneal easily during ligation. 30. Which of these is a commonly used host organism in recombinant DNA experiments? A. Saccharomyces cerevisiae. B. E. coli. C. Agrobacterium tumefaciens. D. All of the above. Answer, D. All of the above. Different hosts are used. E. coli for bacteria, Agrobacterium for plants, S. cerevisiae for eukaryotic systems. 31. Which feature allows plasmids to replicate independently inside bacterial cells? A. Orisite. B. Selectable marker. C. Sticky end. D. LAC-Z gene. Answer, A. Orisite. The origin of replication initiates plasmid DNA replication within host cells. 32. The first genetically engineered vaccine was against A. Rabies. B. Hepatitis B. C. Polio. D. Influenza. Answer: B. Hepatitis B. The recombinant hepatitis B vaccine was produced using yeast cells. 33. The enzyme used to link DNA fragments is derived from A. E. coli. B. T4 bacteriophage. C. Agrobacterium. D. Thermus aquaticus. Answer: B. T4 bacteriophage. T4 DNA ligase from bacteriophage is commonly used in ligation reactions. 34. The first cloned animal, Dolly, was produced using A. Recombinant DNA. B. Somatic cell nuclear transfer. C. Microinjection. D. Gene gun. Answer: B. Somatic cell nuclear transfer. Dolly was cloned in 1996 by transferring nucleus from a somatic cell to an enucleated egg cell. 35. The process of creating identical copies of a gene or DNA sequence is called A. Translation. B. Gene cloning. C. Transduction. D. Hybridization. Answer: B. Gene cloning. Gene cloning produces multiple identical copies of a specific DNA fragment. 36. The TI plasmid from Agrobacterium tumefaciens causes A. Crown gall disease. B. Tobacco mosaic. C. Rust disease. D. Powdery mildew. Answer: A. Crown gall disease. TI plasmid transfers tDNA into plant cells causing tumor-like growths. 37. Recombinant DNA molecules are also called A. Hybrid DNA. B. Transgenic DNA. C. Mosaic DNA. D. Chimeric DNA. Answer: D. Chimeric DNA. The combination of DNA from two sources is termed chimeric DNA. 38. Which step in RDT uses antibiotics for selection? A. Isolation. B. Ligation. C. Transformation. D. Screening. Answer: D. Screening. Antibiotic resistance markers allow only transformed cells to grow during screening. 39. The foreign DNA introduced into host cell is replicated because A. It integrates into host chromosome. B. Vector provides oocyte. C. Host provides replication machinery. D. All of the above. Answer: D. All of the above. The vector replicates using host machinery, aided by ORI sequence. 40. Which of these is a product of recombinant DNA technology used in therapy? A. Interferons. B. Human insulin. C. Monoclonal antibodies. D. All of the above. Answer: D. All of the above. All these biologics are produced using recombinant DNA technology. 41. Golden rice was developed to combat deficiency of A. Iron. B. Protein. C. Vitamin A. D. Vitamin C. Answer: C. Vitamin A. Golden rice is genetically modified to produce beta-carotene, a precursor of vitamin A. 42. The enzyme used to synthesize complementary DNA, cDNA, from mRNA is A. RNA polymerase. B. Reverse transcriptase. C. DNA ligase. D. DNase. Answer: B. Reverse transcriptase. Reverse transcriptase forms DNA from RNA templates. 43. Which is not a direct application of recombinant DNA technology? A. Production of vaccines. B. Gene therapy. C. Protein synthesis. D. X-ray crystallography. Answer: D. X-ray crystallography. X-ray crystallography is used to determine structure of macromolecules, not gene manipulation. 44. The role of selectable marker is to A. Identify transformed cells. B. Cut DNA. C. Bind DNA fragments. D. Replicate DNA. Answer: A. Identify transformed cells. Markers like antibiotic resistance genes help detect successful transformants. 45. Biolistics refers to A. Use of electric current to insert DNA. B. Use of microprojectiles to deliver DNA. C. Use of viruses to insert genes. D. Use of liposomes for transfer. Answer: B. Use of microprojectiles to deliver DNA. Gene gun technique, biolistics, shoots DNA-coated particles into plant cells. 46. Which enzyme is used in PCR and withstands high temperature? A. TAT polymerase. B. DNA polymerase I. C. Ligase. D. Reverse transcriptase. Answer: A. TAT polymerase, derived from Thermus aquaticus. It resists denaturation during PCR cycles. 47. Recombinant human insulin was first produced by A. Genentech. B. Pfizer. C. Eli Lilly. D. Roche. Answer: A. Genentech. Genentech developed the process, Eli Lilly marketed it as Humulin. 48. Which part of the process ensures expression of recombinant gene into protein? A. Promoter and terminator sequences. B. Selectable marker. C. ORI site. D. Restriction enzyme site. Answer: A. Promoter and terminator sequences. Promoter enables transcription initiation, terminator ends it, ensuring expression. 49. The main advantage of recombinant DNA vaccines is: A. Require boosters. B. Use live pathogens. C. Highly specific and safe. D. Complex storage. Answer: C. Highly specific and safe. Recombinant vaccines use purified antigens, reducing side effects and contamination risk. 50. The overall aim of recombinant DNA technology is to: A. Destroy harmful genes. B. Manipulate genetic material for human benefit. C. Produce mutations. D. Study fossils. Answer: B. Manipulate genetic material for human benefit. The core goal of RDT is to modify DNA to produce useful products like medicines, crops, and enzymes. Thanks for watching.